Unlike the sperm, which is very streamlined, the oocyte is very large. As it develops, the oocyte undergoes asymmetric cell divisions to produce polar bodies. And in these divisions, the developing oocyte retains the majority of the cytoplasm. Oocytes also synthesize yolk proteins, and these in many species are going to serve to nourish the developing embryo. The accumulation of cytoplasm in the oocyte results in a cell that is much larger than the sperm. For example, the sea urchin egg has about 10,000 times the volume of the sea urchin sperm. And this picture of the sea urchin egg and the sperm surrounding it helps us appreciate that difference in size. Among the components present in the oocyte are nutritive yolk proteins, and these are more abundant in organisms that have external fertilization and which lack a larval stage. In the case of the sea urchin, the organisms produce a larva, so although their fertilization is external, their eggs are isolecithal. Other components of the egg include ribosomes and tRNAs, and those are going to be needed for the burst in protein synthesis that occurs right after fertilization as the egg's metabolism is activated. Maternal mRNAs are also present in the oocyte, and in many species, these include morphogenetic determinants. These morphogenetic determinants are usually transcription factors, and recall that these are deposited as mRNAs into the oocyte, and their translation is repressed until either ovulation or fertilization. Finally, the egg may contain protective chemicals, and this is particularly important for eggs that are fertilized outside of the body. Some of these protective chemicals are going to make the egg more unpalatable to predators. But there are also protective chemicals that are antibodies or antimicrobial peptides that will help to protect the developing embryo from infection. And this is something that has been described in birds. In most species, the sperm enters the egg before meiosis is completed. As you may be aware from prior courses, in most, but not in all mammalian species, the oocyte is in the second stage of metaphase at the time of fertilization. And once fertilization happens, it needs to complete this meiotic division before the nucleus can fuse with the sperm nucleus. However, this is not the same for all organisms. In some organisms, like the sea urchin, meiosis is completed prior to fertilization. But in the majority of organisms, as you can appreciate from this figure, meiosis is completed after fertilization takes place. In fact, in some species, like dogs, it's the primary oocyte that fuses with the sperm. So this cell hasn't even undergone any reductions in its genome prior to fertilization, and these reductions happen after the sperm enters the egg. I'm not going to ask you specific details about which stage of meiosis the egg is in at the time of fertilization for a particular organism. You should instead know the general concepts, which are that for most organisms, sperm entry occurs before the egg nucleus has completed meiosis, that the specific stage of meiosis at which an oocyte is fertilized varies across species, and that the oocyte has to complete meiosis before the sperm and the egg pronuclei can fuse. Let's now look at the morphology of the oocyte. The oocyte, like any cell, has a cell membrane. But the point I want to emphasize here is that there are features in that membrane that aid in the fusion of the sperm and the egg. And as we'll see later, there are also features that prevent the fusion of multiple sperm with one egg. Among the features that are thought to promote the fusion of the sperm and the egg are microvilli, and these are finger-like projections in the membrane that are made by actin filaments. Another feature of the membrane that aids in fertilization is the presence of receptor proteins, which we'll discuss in a moment, and these help the sperm to attach to the egg. There are also transport proteins that help to regulate the flow of ions across the cell membrane. And of course, that's true of any cell. But in the case of the oocyte, and particularly oocytes of species that have external fertilization, that flow of ions is important to ensure that only one sperm fuses with the egg. And we'll examine this in more detail in a moment. Surrounding the membrane of the egg, there is an extracellular matrix, which in invertebrates like the sea urchin is called the vitelline envelope. And this is what's being pulled away from the egg here. The vitelline envelope contains glycoproteins, and it adheres to the cell membrane by proteins, including glycoproteins, that project from the cell membrane. As we'll see, the vitelline envelope is important for species-specific fertilization. In other words, it helps to prevent interspecies fertilization. And that's important in organisms like the sea urchin, which have external fertilization, because the environment is shared by gametes of different species. Surrounding the vitelline envelope, there's another coat of glycoproteins that's known as the egg jelly. And this is present in the eggs of many species, but it's not present in mammalian eggs. The egg jelly, which is not shown in this figure by the way, has peptides that diffuse away from it and attract sperm and activate it. It also contains sulfated polysaccharides, and I'm going to talk more about this later, but these polysaccharides trigger the acrosome reaction, which is a process that involves, among other things, the release of digestive enzymes from the sperm's acrosomal vesicle, and those enzymes help to digest the path for the sperm to reach the egg. In mammals, a structure that plays a similar role as the vitelline envelope is the zona pellucida. So this is the extracellular matrix of the mammalian eggs. Surrounding the zona pellucida, instead of an egg jelly, there are actually cells here, and these are known as cumulus cells. These are follicular cells that nourish the egg during its development. And there are multiple layers of these cumulus cells, with the innermost layer, the one that is closest to the zona pellucida, being called the corona radiata because it looks like a radiant crown. So far, we've looked at the external morphology of the egg, but within the egg, there are important structures as well, and there are some that actually play a very active role in fertilization. In most cells, not just in oocytes, right under the cell membrane, there's a gel-like cytoplasm that's known as the cortex. And it's composed of a meshwork of actin filaments. And these actin filaments are also involved in the formation of the microvilli that project from the cell membrane. As I mentioned previously, these microvilli are thought to be important in helping the sperm to bind to the egg. In the region of the cortex of the oocyte, there are granules present that are known very appropriately as cortical granules. Like the acrosomal vesicle, the cortical granules are also derived from the Golgi. Another similarity between the cortical granules of the egg and the acrosomal vesicle of the sperm is that they contain digestive enzymes. Differences between the acrosomal vesicle of the sperm and the cortical granule of the egg include the number of these that are present in each cell and the function of the contents contained within each one. In terms of number, the sperm has just the one acrosomal vesicle, but an oocyte can have thousands of cortical granules, and in the sea urchin, for example, there are about 15,000 cortical granules in one oocyte. In terms of function, whereas the contents of the sperm's acrosomal vesicle are going to help the sperm to penetrate through the outer coatings of the egg, the contents of these cortical granules kind of play sort of an opposite role because they're going to help to prevent the fusion of multiple sperm with the same egg. So remember that in the case of the sperm, the enzymes that are released from the acrosomal vesicle are going to help the sperm to digest the path through the egg's outer coverings. But in the egg, the cortical granule releases proteolytic enzymes and mucopolysaccharides that are going to help remove additional sperm that may have attached to the egg at the time of fertilization. The granules also release adhesive glycoproteins and hyaline, which will help to support the blastomeres during early cleavage.